Okay, ladies and gentlemen, the webinar is going to start immediately. To all participants, please use the virtual background and fill the attendance form by the link provided by the committee in the chat box. We will start the session. Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Warm greetings to all participants joining us today. It is a great pleasure to welcome you to the Friday Scientific Sharing Seminar Series 40, organized by the Research Center for Genetic Engineering BRIN today, Friday, August 15th, 2025. Allow me to acknowledge our distinguished guest, Dr. Andri Fadila, Head of Planning and Program Team, Research Center for Genetic Engineering BRIN, to represent Head of Research Center for Genetic Engineering BRIN. Our speaker, Dr. Aswini Rangayasami, postdoctoral researcher at the Research Center for Genetic Engineering BRIN. Our moderator, Dr. Wahyuni, researcher at the Research Center for Genetic Engineering BRIN, and all participants joining us via Zoom. I'm Andulufi Arini, your MC for today's event. We are grateful to God Almighty for the opportunity to gather virtually in good health and spirit to share knowledge and insights this afternoon. Today's seminar will focus on the topic Phytochemical analysis and green synthesis of metal oxide nanoparticles using medicinal plant characterization and biological evaluation. Ladies and gentlemen, to officially begin our event, we would like to invite Dr. Andri Fadila, Head of Planning and Program Team, to represent Head of Research Center for Genetic Engineering BRIN to deliver his opening remarks. Dr. Andri, time is yours.
Okay, can you hear me loud and clear?
Yes, can hear you.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and welcome to all participants for joining us today at the Friday Scientific Sharing Seminar. I am truly delighted to welcome all of you to this wonderful event, which was part of our ongoing efforts at the Center for Genetic Engineering to share knowledge and advance research in the field of science. Before we begin, I would like to extend an apology on behalf of Dr. Rekayasa Mananingrum, the head of the Center for Genetic Engineering. Unfortunately, due to her prior commitments, she is unable to join us today to open this seminar. However, she has entrusted me to convey her warm and greeting and extend her best wishes for the success of this event. And I would like also to express my gratitude to Dr. Aswini Rangayasami, I hope I spelled it correctly, who has kindly agreed to be here today to share her knowledge and experience with us in her presentation titled Phytochemical Analysis and Green Synthesis of Metal Oxide Nanoparticles Using Medicinal Plants, Characterization and Biological Evolution. This is an incredibly interesting and relevant topic, considering the importance of medicinal plants in modern technology, and also for healthcare and development, as well as the significant potential of green synthesis in the field of nanotechnology. Today's seminar is also an excellent opportunity for all of us to learn more about this research being conducted in the field of genetic engineering, particularly focusing on synthesis of metal oxide particles. So by the end of the seminar, I hope all of the participants can also discuss with Dr. Aswini regarding this research. So before I conclude my remarks, let me maybe I will ask you to have more discussion by the end of the seminar, so we can all learn together. And once again, thank you for joining us in this Friday Scientific Seminar. We hope that this seminar brings us great benefits to all of us. And now, without further ado, let us listen to Dr. Aswini for her presentation. And I wish you all engage for fruitful discussion. Thank you.
Okay, thank you, Dr. Andri, for the remarks. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. I will start count from the first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. We will now proceed to the main session, the presentation by Dr. Aswini Rangayasami, which will be moderated by Dr. Wahyuni. Dr. Wahyuni, please take over the session.
Yeah, thank you, Mbak Andalusi, for the opportunity. Selamat siang, Bapak, Ibu, dan teman-teman semua. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Today we will hear a presentation from Dr. Aswini Rangayasami with the topics of phytochemical analysis and green synthesis of metal oxide nanoparticles using medicinal plant characterization and biological evaluation.
Iya, yang ada aja boleh.
Sebelumnya ada yang share screen ya, Mbak Ai Hertati.
Oh, emang iya?
Uh,
Saya share screen.
oh bukan ya. Soalnya kelihatan speakernya Ai Hertati. Wow. Oke. Okay, before we begin, I will read her curriculum vitae. Aswini Rangayasami earned a PhD degree from Periyar University, India, and now she joined postdoctoral program in Research Center for Genetic Engineering. At the moment, she joined the Gates Foundation Banana Project with the similar topics. It's about the metabolomic analysis of the banana, the commercial banana, and also the wild banana, Musa acuminata. And she already published several articles, review articles, and then research articles in high impact journal. And I hope that by joining Banana Project, she could also contribute to publish some publication in a high impact factor journal. Okay, without further ado, I will let Aswini to join with us. Aswini?
Yeah, Professor, thank
Okay.
you.
Aswini, please, the time is yours. Please share your screen and then you can share your previous research during your PhD or your work in India. Okay, the time is yours, Aswini, please.
Yes, Professor, thank you. Professor, if my slides are visible?
Yes, I can see your slides. Okay.
Yeah. Okay. First of all, I sincerely thank the organizer for giving the opportunity to present my research today. I'm Dr. R. Ashwini, currently working as a postdoctoral fellow at the National Research Center and Innovation Agency, Indonesia, under research supervisor Dr. Rayuni. My presentation today focuses on the bridges of traditional knowledge and the modern nanotechnology about phytochemical analysis and green synthesis of metal oxide nanoparticles using medicinal plant characterization and biological evaluation. As we know, the medicinal plants have been used for the centuries as a source of the healing process, and in recent years, all the medicinal plants used for the emerged as eco-friendly biofactories for synthesizing nanoparticles, avoiding the need for chemical and high energy process. In this talk, I will walk you through the phytochemical profiling and the synthesis of nanoparticles and their physiochemical characterization and their evaluation for their biological applications. I began the academic path of the PSc Botany from Sri Sharda College for Women, and the project focused on the types of irrigation. And then I pursued MSc Botany at Periyar University and working on the effect on EMS on plant growth and molecular variation in green gram. And this focused on the phytochemical bioactive compound. This one, I think. This one, you need to switch on the phone. Yeah, because we cannot see your video. Okay. Okay. Well, and thank you for waiting. I'm from Periyar University, India, and working on preliminary phytochemical, antibacterial, and anti-cancer activity on Spermacosia ispida and Heliotropium indigum. The research at the same line, I'm working in the PhD botany and focusing on the studies on Laboratoria revoluta, mediated metal oxide nanoparticle synthesis, characterization, and biological properties. Introduction, medicinal plants played an important role in the treatment of disease and health disorder for the thousands of years. Because of it is the evaluation, phytochemical is one of the main tool for the assessment of the quality of plants for the further biological activities. It is very rich in antioxidant, vitamins, and bioactive compounds promoting the health because it contains the alkaloids, flavonoids, phenols, and so many secondary metabolites components. In these studies, we focused only on the Laboratoria revoluta medicinal plants. And nanotechnology nowadays, it is very important and it is all focusing in the for the biological activities using the green chemistry approach because of the all nanoparticles having a unique physical, chemical, and biological properties. Among the various synthesis of nanoparticles, the green chemistry it is focused for the eco-friendly and the mainly cost-effective nanoparticle synthesis. There are various metal oxide nanoparticles having a unique physical, optical, magnetic, catalytic properties, making the ideal for biomedical and various applications like silver, gold, titanium, bismuth, so many metal oxide nanoparticles, including the applications of medicine, agriculture, environment remediation, and catalysis. Objective of this study collected the plant and it is authenticated by the BSI Coimbatore and separate the phytochemical by the TLC analysis for using a Laboratoria revoluta bulb extract and determine the phytochemical components from Laboratoria revoluta bulb extract using GCMS analysis and synthesized to the silver and titanium nanoparticles, and it is characterized by the UV visible spectroscopy, XOR, the FTA or TEM and the EDOX nanomaterials. Evaluate the biological activities such as antibacterial, anti-cancer, and antioxidant activities. Investigate the larvicidal activity, histopathological alteration against Aedes aegypti, and study of the toxic effect of plant-mediated nanoparticles using Artemia naphthalene. These are the work done by some of the Laboratoria species, Vultifolia, Gramnifloria, Floribunda, Hyperbundans, and Revoluta, Scalioleus. These some of the species of bulb and leaves are mostly actively involved in the phytochemical screening, somatic embryogenesis, and the novel homoisoflavonoids antioxidant activity. It consists of the homoisoflavonoids, and it is act as antioxidant, anti-cancer. So this was the
Aswini,
review.
sorry for the interruption.
Professor.
Could you speak slowly a bit
Okay.
because we are Indonesian,
Okay,
so
Professor.
it's not native. So please.
Okay, Professor. I'll
speak
follow that.
very slow.
So based on this review, the Laboratoria species having a high rich bioactive compounds to analyze the biological activities, materials and methods. The plants were collected and identified by the Flora of Presidency of Tamil Nadu, and it was authenticated by the Botanical Survey of India at Coimbatore. Preliminary phytochemical analysis, it is the main used to analyze the plant to what are the chemicals present in our samples. So based on this, Brin and Turner, 1975 is what followed for the analysis. Alkaloids, flavonoids, phenols, terpenoids, saponins, tannins, carbohydrates, and so on. We have to analyze the qualitative analysis by the TLC analysis present in the
Maybe host will try to mute Mbak Nurul ya. Mbak Nui,
Enggak,
on.
salah. Jadi yang setting. Tapi kalau misalnya nggak benar jangan.
Okay, Aswini, just continue.
Okay, sure. To identify the presence of different phytochemical compounds in the plant extract, we carried out the qualitative analysis by TLC analysis using silica gel plate method, following by the Priyanka et al. 2013. But we can identify the volatile and non-volatile compounds present in the extract using by the GCMS analysis, followed by the Arun et al. 2010, like alkaloids, flavonoids, terpenes, tannins, and carbohydrates. Synthesis of nanoparticles. We synthesized silver and titanium dioxide nanoparticles using a green approach chemistry when the plant extract the combined with the both reducing and stabilizing agent. 1 m and 5 mm of silver and titanium dioxide mixed within the plant extract, aqueous extract, and it is mixed within using a magnetic stirrer, and it is kept into the dark condition for the silver nanoparticles, and it is turned into a dark color. It is presence of silver nanoparticles, and the white precipitate, it is confirmed the presence of titanium dioxide nanoparticles. This is both are characterized by the UV, FTR, XRD, HRTM, and EDOX analysis, and it is applied for the various applications. Antibacterial and antifungal activities were analyzed by using a well diffusion method, and antioxidant activity is analyzed by using a DPPH and ABTS analysis assays. We evaluated the anti-cancer activity against MCF7 used in the different concentration of a nanoparticle extract and the plant extract. The MCF7, it is treated with different concentration. It is the assessment based on the mitochondrial activity, and when the yellow MTT dye is reduced to purple formations of the viable cells. The larvicidal activity was assessed by the using of Aedes aegypti, and the third instant larvae were used for these studies according to the WHO 2005 protocol. Next, histopathological studies, how the nanoparticles involved and into the cell death into the plant. So according to this, Bhanumati et al. 2017 was followed for the histopathological studies. The larvae are induced into the stain and sectioning, and staining by the hematoxylin and eosin stains were used. And finally, the toxicity studies were analyzed by using Artemia species for non-toxicity studies using by our extracts. Next, we go to the results. Phytochemical screening, TLC, and GCMS analysis. The preliminary phytochemical screening evaluated by the chloroform, ethyl acetate, and methanol extract using these three solvents. Among these three, methanol extract showed the highest compound of present secondary metabolites like alkaloids, flavonoids, saponins, and oils and resins, carbohydrates. Next, the chloroform having a moderate presence of the secondary metabolites like alkaloids, saponin, and carbohydrate, tannins, and oils and resins, compared to these two, ethyl acetate having the lowest secondary metabolites activity. TLC analysis, it is used to analyze the presence of preliminary phytochemicals. What are the present compounds in our presented samples? The slides were evaluated under the normal and UV and iodine vapor light. We got the 8 fractions from this TLC plate. And the 8th fraction is having a high amount of presence of secondary metabolites in all the 3 extracts. Among these, the methanol extract plant pulp of it is having a high amount of the components. Next, we go to the quantitative analysis for Laboria revoluta bulb. Among these 3 extracts, we analyzed the methanol extract having a high polarity, so it having a high components, alkaloids, flavonoids, saponins, tannins, and carbohydrates. Among these, saponins and carbohydrates have a highly presented in the methanol extract. Chloroform having a moderate mid polarity of chemicals. So these components having a moderate level of the secondary components. This is the GCMS chromatogram of the chlorophyll, ethyl acetate, and methanol extract. Among these three, methanol having a high number of primary phytochemical components, secondary metabolites among presenting the methanol extract. Totally 17, 20, and 36 components present in the methanol extract. These components are highly involved in the many biological activity like N-hexodeconic acid, oleic acid, and octogosane, and many number of secondary metabolites derived from these plants, and it is actively involved in the antibacterial, anti-cancer, antimicrobial. So from these results, we have Ledoporia revoluta having a high rich bioactive compounds, and it is leading to produce in our research for biological activities. Characterization of silver and titanium dioxide nanoparticle. UV visible spectroscopy, it is used to analyze the synthesis of silver and titanium dioxide nanoparticles. Predominantly, the surface plasmon resonance peaks at for silver synthesis 472 nanometer, and for titanium dioxide, the peaks is 323 nanometers. These peaks are confirmed the presence of synthesized silver and titanium dioxide nanoparticles. This is XRD patterns of the silver and titanium dioxide nanoparticles. This is the XRD patterns of silver nanoparticles to theta values of 29, 33, and it is correlated to the corresponding to crystal planes 210, 112, 111, 123, 140 according to respectively. The 111 strong peaks, it is confirm the presence of silver nanoparticles in our samples. Then this peaks for the titanium dioxide nanoparticles, and this is for the corresponding crystalline planes 101, 103, 113, and 105 respectively. This is the confirm the anatase phase of the titanium dioxide nanoparticles. And this spectra of FTAR of green synthesized silver nanoparticles transmission peaks. This is corresponding the presence of vibration of OH, the phenolic group, and aliphatic amine and bending amine groups like this. And this is for titanium dioxide FTR spectrum of our sample for peaks, and it is corresponding to the alcohol group, aliphatic group, aldehyde group, and carboxyl becomes OR. These spectrum of confirmed the presence of functional groups in our sample synthesis. It is synthesized silver and titanium dioxide nanoparticles. Next, HRTM of the synthesized silver and titanium dioxide nanoparticles. It confirmed the size and shape of the nanoparticles. For the silver nanoparticles having a sphere, and the average size is 30 nanometer. And for the titanium dioxide nanoparticles, the average range is 85 nanometers, and it having a spherical in shape. And this is EDOC spectrum of the synthesized nanoparticles. The highest peak of silver nanoparticles and additional within the copper and carbon. This peak is confirmed the purity of the silver synthesized in our samples. Then this is for the titanium dioxide nanoparticles EDOC spectrum. This spectrum, it is predominantly high peak. It is represented the presence of titanium dioxide nanoparticle, additional peaks within the minor peak in the oxygen and the carbon dioxide. These peaks are highly evaluated and concluded the presence of purity of presence of silver and titanium dioxide nanoparticles. Next, move to the biological activities and antibacterial activity. We can use both the gram-positive and gram-negative bacteria for silver and titanium dioxide compared within the methanol extract. Among these four bacterial pathogens, using well diffusion methods, and we can use the chlorophynical used as a standard solution for this activities and under nutrient agar medium. Among these, the synthesized silver nanoparticle extract showed the highest zone of inhibition in the Clostridium tetani. This is the high zone of inhibition compared to titanium and the methanol extract. And the very least zone of inhibition present in the Vibrio cholerae in methanol extract. Antifungal activity. This is the pathogens used for in these studies used methanol, silver, and titanium dioxide extract. Among these fungal, the silver nanoparticles extract showed the highest zone of inhibition in the Niger family species, 6.23 mm at the 25 micrometer, comparing to the methanol and the titanium dioxide nanoparticles. This is the general mechanism for these antibacterial activities for the general nanoparticles. Silver nanoparticles, once the nanoparticles enter into the bacterial cell and it is membrane damage, it damage the membrane and leakage into this, and it leak and release the ROS ions, and it is bind to proteins, DNA, and it inhibit the cells to the function. It is generate the oxidative stress and cell death. Antioxidant activity using the DPPH and the ABTS assay. For DPPH, we can use the methanol, silver, and titanium dioxide nanoparticles. Among these silver nanoparticles showing a high DPPH activity against DPPH, 79.73 IC50 value. Next, the methanol extract showed the moderate activity at the IC50 value 92.61 against the DPPH scavenging activity. TiO2 nanoparticle, it showed the highest IC50 value 97.27, but it confirmed the lowest value of activities of DPPH radical scavenging activity. Among at the same for the APTS radical as scavenging activity, the synthesized silver nanoparticle showed the highest activity at the lowest IC50 value of 75.84%. Next, anti-cancer activity. Anti-cancer activity, we have analyzed the same extracts were used against MCF cell line. MCF cell line we can use collected from the Madras Research Center for the Entomology. And these they showed the among these three, silver nanoparticle showed the lowest IC50 value of 43.29 microgram per ml compared to other two extracts. It showed the highest cell death for the anti-cancer against the MCF7 cell line at the highest concentration. This is the dose-dependent activity, and when increasing the concentration of the nanoparticles, the activities are also increased for the cell death. Larvicidal activity. We can use to the Aedes aegypti larva at the third stage larva. We can collect from ICMR and Madurai. The cysts were collected and growed, and the third stage larva are treated within the different concentration of methanol, silver, and titanium dioxide nanoparticles. Among these, silver nanoparticles showed the highest larvicidal activity against Aedes aegypti at the lowest 14.14 LC50 values, and against LC90 51.91 milligram per ml values, compared to the methanol, it having a moderate value at the 28.82 LC50 value, and the LC90 value is 81.01. Titanium dioxide showing the minimum activities comparing to the other activity. And we have to analyze evaluate the how the nanoparticles detach the Aedes aegypti larval into the cells. Nanoparticles adhere the larval cuticle, penetrate the tissue, and generate the ROS damaged cell membrane, and interface with the enzyme. It causes the mortality rate, and it is reduce the epithelium damage, and the interference siphons are detached within the plant body. And this compared to titanium dioxide, the cell membrane and the penetrating the epithelial cell, it is very high, and it is cell death in parts are very high in the silver silver nanoparticle synthesis at the 30 mg per ml concentration. It's nanotoxicity. We generate the toxicity level checked using the Artemia napoli. Artemia napoli, it is the zooplankton. This is used for mainly for non-toxicity study. And we have focused in the silver and titanium dioxide nanoparticles for hatching, motility, and morphological changes of the Artemia napoli species. The hatching percentage, when the concentration increase, the percentage of mortality and hatching percentage or increase with the percentage of 88 for the silver nanoparticle for hatching percentage, and the 13% mortality for in the silver synthesized nanoparticles extracts. And for the titanium, it is a comparatively silver, it is less activity at the 86% of hatching and the 12% of mortality rate of on Artemia napoli. From the morphological changes, there is the nil changes for in the control. We have to use the water and sieve pond water and combine together. And compared to silver and titanium nanoparticle extract, the silver nanoparticle showed the minimum less eyeball in the morphological changes comparing to the titanium extract at the 12 ppm when increasing the concentration. And summary and conclusion. 3 solvent extracts from Laboratoria revoluta bulk were screened, and methanol extract showed the highest phytochemical diversity. Alkaloids, flavonoids, saponin, carbohydrates, and oils and resins were selected for further study. So silver and titanium nanoparticles were green synthesized, and it is characterized was confirmed by in the size and shape is UV, XRD, FTER, and EDOX. The size for the silver nanoparticles 13 nanometer, and the titanium dioxide for 85 nanometer, and spherical and sphere shape for the nanoparticles. Among the silver nanoparticles having the strongest antibacterial against Clostridium tetani and Aspergillus niger, and it is against the highest antioxidant activity against DPPH at the 17IN and ABTS for 75 says against that two antioxidant activities. It is also against the cytotoxicity MCF7 cell line, breast cancer cell line within the IC50 value of 43.29 microgram per ml. And it is very potential activity against the chikungunya and Zika, and this cause the vectors by the Aedes aegypti. It is against the causing of mid-cut epithelial damage in the larvae. It is the slightly toxicity at the higher concentration when the silver nanoparticle extract, and at the Artemia napoli at the higher concentration 40 to 55% mortality. It is eco-friendly, but it is effective biocontrol potential. In a nutshell, it is observed from obtained the data was clearly reported that the methanol extract Laboratoria revoluta bulk was rich in bioactive compounds, and it is used to green synthesized silver and titanium dioxide nanoparticles with a strong antimicrobial, antioxidant, and anti-cancer against MCF7 and antioxidant activity against the DPPH and ABTS. It showed the highest efficacy and eco-friendly potential for disease vector control and biomedical applications. My present study, comparative metabolic analysis, reveals quality profile of banana varieties. Objective of my study is to extract the secondary metabolites from different varieties of commercial banana fruits to analyze the secondary metabolites of phenolic flavonoids and polyphenols in commercial banana varieties using HPLC GCMS analysis to characterize the volatile compounds contributing to the aroma profiles, to analyze the pigment responsible for the color of banana fruit, to identify and qualify the key metabolite associated with the sweetness in banana fruit. And once again, thank you for giving this opportunity.
Okay.
Okay, Aswini, thank you very much for your presentation. It's huge data actually, yeah, and then lot of data, and it's for me, it's hard to digest. I mean, because we need to how to say that, carefully listen and then pay attention to your slides, each slides, because it's one slide to another slide is like a related. But I have also some questions that it's only confirmation questions. But before that, I will ask the audience first if they have any questions or maybe in the chat. Oh, okay. Maybe the audience here would like to have questions to Aswini. Please use the raise hand feature for that. Okay, while waiting, maybe I have some question, Aswini, because
Yes, Professor.
it's too many data that you already did so many experiments for that just to check the bioactivity of the compounds. Yeah. I'm curious about your experiment about the bioactivity. First, you say that you compare the methanol extract and then several solvents, right?
Yes,
For
Prof.
the bioactivity. And then it's showed that methanol extract has had higher bioactivity compared to the other solvents.
Yes, Professor.
Okay.
Compared to ethyl acetate and chloroform solvent.
Okay. Okay. And then you did also the fractionation from them.
From them.
Yeah.
For
And
the
then
and the ethyl acetate extract.
okay. But then for all the analysis for the I saw also for the stream analysis and antimicrobial analysis, did you choose which extracts for the experiment or for the analysis of antimicrobial or
From the biological activity, I have
yes?
selected the methanol extract, silver,
Okay.
and titanium.
But it means that you use all the extract, not the fraction of each fraction.
Methanol extract for biological activities, I can stop from because of the based on the biochemical number of biochemicals activities, I can choose only for methanol extract for the further biological activities compare within the nanoparticles extract.
But then you did the fractionation. So could you explain me what's the purpose of the fractionation?
Fractionation for the analysis of the color identification using by the TLC plate. And this fractions, I have got the 8 fractions. I can stop from that. And generally, I can chromatograph from basic for the methanol,
Could you show
chloroform.
me, please, in the slide, Aswini, just to make it clear? Sorry, yeah.
Yeah, okay, Professor. Okay, sure. Yeah. The arrow marks are represented the fractions of the chloroform, ethyl acetate, and methanol extract presented in my compound extract.
Yeah, but I think you have a slide with the fraction 1 till 8. And then you said the fraction 8 has the, yeah, this one. This is from the TLC.
Yes, Professor. Your for TLC ORF value
Okay.
based on the ORF values.
I see, but you did not do the fractionation with the HPLC. It's only the fraction
Only
from
the
TLC.
TLC.
I see. Then you use all the extract, the methanol extract for the
For the
bioactivity.
bioactivity.
Okay,
Yes.
I get it. Okay, thank you, Aswini.
Thank you.
I saw in the chat room there is a question from Mbak Endah. Aswini, should I read it for you or?
Yeah, Professor, please.
Yeah, thank you for your nice presentation, Dr. Aswini. I want to ask, how is the toxicity, acute or chronic, of the silver or titanium-derived nanoparticle in vivo, for example, in mice?
In mice.
Yes, this is the first question. Yeah, maybe you can answer it first.
Yeah, for mice, the silver nanoparticles, it can easily penetrate because it having a high surface area and the potential of physical and chemical properties, it is high, and the small number of nanoparticle size comparing to the other nanoparticles. So it is easily penetrate into the cell membrane of these cells, and it is damaged the parts, and it can control the growth of the liable cells for the frozen stage, and it is produce the ROS reactions, and like OH radicals, ion H radicals, it can release the ion particles, and it can generate the ROS, and it can damage the cells, and it can cause the growth of the cell membrane. And also it finally it detached, and the death of cause of the death cell. So this is the reason why, because of the based on the nanoparticle size and these high surface stability value for the main reason, and it is easily penetrate into the cell membrane, and it is cause the ROS into the cell damage, and it can finally leading to the cause cell death.
Okay, and there is also a second question from Mbak Endah. Is it possible to generate nanoparticle of extract without the addition of silver or titanium?
Yeah, it is possible to penetrate because we further studied and synthesized the zinc oxide nanoparticles and iron oxide nanoparticles. These are based on their activities of the optical density and the stability concentration of the samples using the mainly it is reliable to the plant phytochemical. So we have to synthesis the green chemistry comparing to the chemical synthesis of nanoparticles.
Okay, Aswini, is it possible to use another element for nanoparticle for making or creating nanoparticle except the silver or titanium?
Yes, Professor, that it is possible. That's why I'm telling the zinc, and even nowadays the bismuth and the carbon coated carbon dots nanoparticles are mostly involved for the binding of the plants, and it is mostly used to do the treatment of environmental remediations for the wastewater treatment. So it is ongoing the process, and also it is applied for in the cancer studies focused on the liver, and nowadays the breast and the liver cancer is causing death leading. So both the biological and the environmental wastewater treatment and hospitality water also we have checking the for the analysis to curing the toxicity in the water level.
Okay, but the element for the biomedical application and environmental application has the differences. I mean, for example, for my biomedical is safer using the silver or titanium. Is it like that?
For biological, mainly in the
Yeah.
concept for comparing to the silver and titanium, mostly used for the silver nanoparticles from the beginning, but nowadays the number of nanoparticles based on their activities were used for the cancer studies.
Okay. Mbak Endah, Mbak Septi has Aswini do you have any questions for Aswini again, or is it okay for her answer?
Maybe one more question.
Okay.
So, Dr. Aswini, do you think it is safe for animal or for human to take this nanoparticle?
Yeah, that's why we checking the toxicity level according to that based on the using a zooplankton. And some of the dose, some elements are increasing the concentration. While we increasing the concentration, mild changes or interact within the toxicity level. So we can choose from the elements based on the concentration. So that is the best way to the control the concentration and interact within the cells, the live cells.
Yeah, thank you very much, Dr. Aswini.
Thank you, Professor.
Okay. There is also another question from Bapak Nurfianto Giar Pangidung from Malang. The first question is why do you do many studies? Why do many studies on plant-mediated nanoparticle synthesis use medicinal plant extract?
Yeah, medicinal. I have selected the 4 or 5 plants from the my survey of in that time. Mainly medicinal plants having a number of secondary metabolites comparing to the flowering plants like ornamental plants like. So we focused into the already we reviewed that the medicinal plants having a secondary metabolites comparing to the others. So I focused on the medicinal plants. So I have studied already in the 2 or 3 plants in previous my MPhil and MSc study. So it concluded that, and based on that 2 or 3 medicinal plants, and also I have again surveyed and collected the many plants from these medicinal plants. I studied the phytochemical analysis and derived from that. I've chosen one plant. So basically, the medicinal plants having a high number of rich secondary metabolites compound, and it is bioactive compounds involved in the gapping and stabilizing agents for the nanoparticles.
Yeah, and I also think that maybe nanoparticle is one of the method for the application of the medicinal plant extract, yeah, for human, for example, or for the other organism, right? Yeah, just to have a kind of direct target. Okay, the second question is why choose the Ledeboiria revoluta plant in your research? What are its advantages compared to other Ledeboiria species or other medicinal plants? And what new insights or novelties does this plant have, especially for a plant-mediated nanoparticle synthesis?
Yes, I told like that. I surveyed many plants. From that plant, I have chosen a Ledeboria revoluta based on their. I'm reviewed from the forest women's and peoples because of their tailing. This is mainly for in that centuries it used for the bites, snake bites, and it's used for fever. So like that, and it also they analyzed for the phytochemical screening. So based on that, with having that metabolites, so I'm choosing for based on that, and also reviewed the many papers. And this is the first report for using this plant bulb and leaf using nanoparticle extract, and that is mainly focused on the anti-cancer and larvicidal activities.
Okay. And the third question is what marker secondary metabolites found in Ledeboria revoluta in high concentration, and what role do these compounds play in nanoparticle synthesis?
So based on the GCMS and the review, in my plant having the bulb and leaf and the isoflavonoids, it is the unique feature in the Ledeboria revoluta, homo isoflavonoids. That's compound. It is a unique compound for in this Ledeboria revoluta. And this compound also actively involved, I showed the slide, actively involved in the many biological activities like bacterial and fungal and oxidant activities for making this conclude the homo isoflavonoids. It is a unique properties, secondary metabolites of the Ledeboria revoluta.
Okay, so it means that the marker or the highest metabolites or the abundant metabolites in your extract is phenolic compounds or flavonoid, yeah, Aswini.
Yes, Professor.
Okay. And you did the GCMS analysis for that. Is it correct? Am I correct?
Yeah, GCMS, and this is the peak, and get the based on the number of compounds per
Okay.
chloroform,
Yeah.
and this is ethyl acetate, and most of these trees having the isoflavonoid and hexane amine groups, and methanol. For this three extracts, we have to finalize the chemical compounds, identify the biological application. So for this, an hexadeconic oleic acid and already reviewed, based on the reviewed, the isoflavonoids and homoisoflavonoids are this mainly used in this for actively involved in the biological activities. I already showed in this slide.
Do you have any reasons why do you use GCMS analysis rather than HPLC? Because maybe is it what you did? Is it was it profiling or you already do? I mean, you already known before that you have this compound in your extract.
Yeah, I can stop the GCMS not for the HPLC based on the reviewed, and it is concluded by the presence of only TLC GCMS analysis based on this result.
Okay. But for the main compound of the, for example, the flavonoid itself, have you already known before? I mean, for the GCMS data, you can only like have a fraction of the volatile part of the compound, right? But
Yes,
the real compound, do you already know? Did you
I
already know?
No, I did not derive any particular compounds. I can only used in the extract for the TLC and GCMS analysis. And after getting this only, I have know this homoisoflavonoids present in this, because but this compound is already reported in one of the author, and this is the unique compound present in this, novelty compound present in this Laboria revoluta. So based on that reasons, I have chosen for the Laboria revoluta, not only the flavonoids, it having a many number of compounds present in that pulp and leaf parts.
Okay. Yeah. Okay. We still wait for the questions from the audience. Please, if you have questions.
Can I ask a question?
Yes, please.
Okay. Thank you for your presentation, Dr. Aswini. So I'm curious about how do you ensure the consistent phytochemical profiles for reproducible nanoparticle synthesis, since we know that, you know, there are several things that can affect the production of secondary metabolites, right? Such as season, location, cultivation method. So, and each of this thing can affect the production of the secondary metabolites. So how do you ensure that the profile, the consistent profile of your nanoparticles? Hope you catch my question. Thank you.
I can profile my phytochemical screening based on this plant using a TLC and GCMS. So they're based derived from the compounds. What I have to get that compounds, it is actively involved. Though it is bind within the nanoparticles and easily for in the gapping agent, stabilizing agent. Basically, the plant metabolites actively bioactive compounds. All the medicinal plants having that compound, but then the percentage of compounds are in the involved for in that reaction for the gapping and stabilizing agent for the nanoparticles. So based on these fractions, and I have concluded that this plant having plant secondary metabolites reaching this highest amount it is involved for in the my further activities. So I have chosen for that.
Ya, Mbak Aini.
Okay, thank you for the answer.
Oke,
Thank
thank
you.
you Mbak Aini. Aswini, I'm curious about your methanol extract. Some time ago during my study, there are some groups also doing this bioactivity analysis using the different kind of solvents, and then they check for the toxicity of methanol itself for the cell culture. And then did you also check that? And then because at the end they have like the percentage of the methanol that cannot harm the cell culture.
For toxicity, I have been used only synthesized silver nanoparticle extract. Um, but I cannot check. Professor, I have used only for synthesized silver nanoparticle extracts for the toxicity study. Because of the I can derived from that based on the low versatile activity, I can stop there in the methanol extract because due to the damaging the Aedes aegypti cells, so I can stop that. The control was is very less, so it affected the larval activity is very less. So I can stop there in that methanol extract for that non-toxicity study. So I go for the nanoparticles extracts only.
I see. So you use different concentration like 3 ppm, 6 ppm, up to 12 ppm of the extract, yeah, for the bioactivity analysis. Okay. I don't see any questions from the chat room. I will still open the question session. Maybe we can wait for 1 minute, Aswini,
Okay,
just
Professor. We will wait.
for the audience. For the application in plant science, do you have any clue about that, Aswini?
Sorry, Professor.
The application of this nanoparticles for the plant science.
Applications?
Yes.
In my study for biological evaluation or
Yeah. And now maybe a future application. Yeah.
yeah,
Using the
yeah, using nanoparticles.
Mhm.
Now we are doing already said like wastewater treatment and hospitality water treatment. And when one of my friends group visited a hospital and collect some water and making a check for and applied for the nanoparticles for mainly for photocatalytic activities and for using the metal oxide nanoparticles like copper, bismuth-based, and carbon dots, many nanocomposites in the form of composites are used for applied for the photocatalytic, mainly for wastewater treatment.
I see. So it's more to environmental experiments regarding to related to plants adaptivity. Yeah.
Yes.
There is one question. Sorry.
Yeah, absolutely. And it also in future trying to apply for in the plant growth like green gram, black gram, and focused on the stress, what drought stress and the water stress level. It combined to nanoparticles. It reduce the stress level among the plants. Evaluations like green gram, black gram, like only focused for in the drought stress.
Yeah, thank you, Aswini. There is one question in the chat room from Pak Nurfianto. He asked, are there specific secondary metabolite compounds in large concentration? Are there specific secondary metabolite compounds in large concentration in the plants as marker in your research? For example, in nutmeg, there is a myricetin, which has a significant influence in pharmaceuticals or traditional health.
So there's a number of metabolite compounds present, but in my plant already told in the homoflavonoids, isohomoflavonoids it's present. But in other components or present in a lot of medicinal plants, mainly it is based on the presence of alkaloids, flavonoids, and phenols and saponins. These the four which focused on the presence of the selectively for the biological activities for mainly focused for the pharmaceutical or in the traditional health, mainly in the alkaloids, flavonoids, and phenol saponins. These components are focused for the presence, and it is in the we have used plant extract that is go for further for the biological activities.
Okay, Aswini, thank you very much for your presentation. And Pak Nurfianto also say thank you for the answers. I think we can finish this session. Thank you, Aswini, for your time, for sharing your previous research project with us. Saya kembalikan lagi ke MC.
Thank you, Dr. Wahyuni, for leading a very interactive discussion. And thank you, Dr. Aswini, for a very interesting presentation. And I would like to remind to all participants, please fill the attendance form by the link in the chat box. Okay, ladies and gentlemen, we have now come to the end of today's Friday Scientific Sharing Seminar Series 40. On behalf of the organizing committee, we would like to express our sincere appreciation to our speaker, moderator, and all participants. We hope today's session has provided valuable insights and inspiration for your research and professional endeavors. Thank you for your kind attention and active participation. See you in the next Friday Scientific Sharing Seminar. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you. Thank you all.
Thank you, Aswini.
Thank you, Professor. Thank you. Shall I leave from the meeting?
Yeah, you can leave the meeting.
Thank you.
Yeah.